Hi students, in this video I am going to explain you about the enzymes. It is most important for the NEET also. And with 3 words I will complete the enzymes. What is enzyme? Enzyme, without enzyme there is no metabolic reaction. First point, remember it. What is metabolic reaction? Continuous metabolic reactions keep the cell in the living stage. So life is depend upon the continuous living reactions, metabolic reactions. Metabolic reaction, there is no metabolic reaction without enzyme. Means you will come to know what is enzyme. That much important. And that enzyme is made up of the proteins. And what it will do? Enzyme is a specialized biological catalyst, and that accelerate the metabolic reaction in the living organisms. Means just I told that is the metabolism reactions will be accelerated, means it will be increased by the catalysis of this enzyme. Enzymes are made up of the proteins except ribosome. Ribosome is the enzyme which is made up of with RNA, and 23 RNA, 28 RNA, it will come in the second year. No problem. That is in the molecular basis of inheritance it will come, but in advance I am telling. So ribosome. So enzyme is it is a catalyst. Without enzyme there is no metabolic activity, metabolic reaction. Without metabolic reaction there is no life. And so enzymes are made up of with proteins, such type of enzyme, but ribosome enzyme is made up of with the RNA. What it will do? The enzyme works as a biological catalyst, and it accelerates, increases the speed of the metabolic reaction in the living organisms. That is the first point. And in this one also bits are there. Remember it. Statements it will come. Next, now the biological catalyst. What is catalyst? Catalyst means any substance that speed up chemical reaction without consumed in the process. What is catalyst? When the reaction is going on, it is a catalyst. What it will do? It will speed up the reaction. The reaction speed will be increased with the help of a catalyst, and whenever it is increasing, it is not involved in that reaction chemically. Means without consumed, means if it is chemically involved, it will become substrate, but it is not like that. Only the thing is catalyst will increase the speed of the reaction. Such type of the biological catalyst is the enzyme. Enzyme is made up of proteins. This is the thing he has given in the book also. Now you see, the enzyme is chain of protein. Protein is it is a chain of enzyme also is a protein. So it is a chain of protein. And here this is the tertiary structure. What is the tertiary structure? How it will be made? In the previous video I discussed. But again, it is the tertiary structure. Tertiary structure will be made by the secondary structures, means that is the alpha helix and beta plated. Means how it is? The thread used to self coil. You studied already. In the self coiling, the thread like structure, it used to self coil as the helical, and then it will be some places it will be the beta plated and the self coil again. Hollow wool ball will be prepared. This is important. Means it is a thread. The thread is extended with some places alpha spiral and alpha helix, and then some places it will be the beta plated, and again this thread will be continued self coiling. So like that it is to prepare, that already I explained in the previous video also, the tertiary structure. Such type of the tertiary structure it is to have. Whenever it is doing all the things, naturally what happens? At this time, protein chain folded, means whenever it is folding, self-coiling, and the alpha helix and beta plated, whenever it is doing, what happens? Fold on itself, it forms the crisscrosses, means it is crossing that one, this one, and a crisscross. Go like this and like this. This is crisscrosses. And whenever it is doing like this, what happens? It is along with the crevices and pockets. Whenever it is going like this, some places crevices like this, and some places it will be like this. This is the cup-like shape. In this one, substrate will come and sit. When, why, how it is forming? Because of the crisscrosses form. In the crisscrosses, there will be the crevices, means like creeks, and some places the packet, like this. So these things happen because of the tertiary structure. Tertiary structure is formed by the secondary structures also. Secondary structures used to present in the tertiary structure. Altogether, they used to form the self-coil and forms the creases and the packets. This is the most important. How the packets are formed? Because of the tertiary structure. Tertiary structure is formed by the alpha helix and beta plated. Altogether, they are self-coiled and forms the creases and packets. Now we'll see what is this packet. This is the most important. The packet formation is the most important. The packet is called active site. This is important. Why the active site? Active site, students used to get the doubt here only. What is the active site? Active site is formed in the tertiary structure of the protein. And tertiary structure made by the alpha helix and beta plated. And this is crisscrosses, the self coil. Allowool ball-like structure is to form, it crisscrosses, it is to form the creases and packets. That packet is called as a active site. So what? In the active site is a cup-like shape. In that one, substrate will come and sit. Means here, you see, this is a cup-like shape. Substrate, like ball, it will come and sit in this. So the cup-like shape, that only, the active site, substrate fits, the substrate will come and fits in the active site, and then the enzyme protein catalyzes. Then the substrate which is going to convert into the product will come and sit in the active site. Active site, how it is formed, I told. And that this, where the active site? In the enzyme. The enzyme is protein, or it is self-coiled. After sitting, the protein will do it. That is a reaction, catalyze. Means the whatever the substrate will be converted into the product with a high speed when the substrate comes and sit in the active site. Where the active site is present in the enzyme. Enzyme is the protein tertiary structure. Understanding now? Now you see here, this is the formation of the active site, most important and highly confusing to the student. For that only repeatedly I am telling. Active site is a packet-like structure which is formed on the tertiary structure of the enzyme protein. Protein is a enzyme protein. On that one, it is substrate used to come and sit in that one. Then the entire protein, that is enzyme, will this catalyzes the reaction. High speed it will do. Now, whenever we are seeing here, now the thing is, there is a lot of difference. Catalysts are present in the inorganic reactions also, the metabolic reactions. Inorganic also, whenever in the industries also catalysts they will use. That what they do to catalyze work efficiency at a high pressure and temperature. The inorganic, whatever the catalysts are there, they will work at high pressure and high temperature also. Its efficiency will increase. High speed it will do, increasing the pressure and temperature. But enzyme is also biological catalyst. This also should increase, no? That actually inorganic catalyst is doing this, but it won't do. This is the important. Why? Because the enzyme is formed by the tertiary structure of the protein. Protein is formed by the amino acids peptide bonds. Now the peptide bonds will break when the temperature increases more than 40. When the temperature increases more than 40 degrees, the tertiary structure will lose its structure. When it is losing the structure, there is no packets, that is active sites. When there is no active site, there is no reaction. That is the he want to tell. So temperature won't affect on the inorganic reactions. Temperature pressure actually it is to increase, but in the enzymes when they are working as the biological catalyst, they will work up to the some extent, that is the 40 degrees centigrade only. After 40 degrees centigrade, they will be denaturated. Means the enzyme will lose its tertiary structure. Because of that one, there is no question of formation of whatever the active sites are there, they also will be denaturated. So it will become functionless. This is most important. He can ask it as a statement or matching anywhere he can ask. Now, you see, however, some enzymes will work at a high temperature also. What are they? That also he mentioned in the. I am going through the NCERT because he is giving the questions in the NEET or anywhere based on NCERT. Now the see. That is at 90 degrees centigrade also some enzymes used to work. Generally, most of the enzymes will be denaturated at 40, but some are working. What are they? The microorganisms which are living in the hot vents and sulfur springs. So this will come clearly how it is in the biotechnology. There you can. But hot springs means the temperature will be very high. In that also some microorganisms used to live. So the microorganisms enzymes are can work at high temperature up to 90 degrees centigrade. That also he has given. That is the thing. Now we will see chemical reactions. Means either inorganic chemical reaction or organic chemical reaction, whatever it is. Metabolic reaction is also nothing chemical reaction. So the chemical reaction, how it will be, that also he explained. So chemical reaction means chemical compounds undergo two types of reaction. Means any chemical compound, what is the entire thing is made up of the matter, and the matter is a chemical compound. So the chemical compound undergoes the chemical reaction, two types. What are they? Physical and chemical. Physical change. Physical change means the ice will melt and forms the water. It is a physical change, state only changing. And if you give the temperature, the water will become evaporate. But composition is H2O only. And the state only means solid to liquid, liquid to vapor, it is changing. It is a physical change. But chemically, it is other H2O. But if it is changing chemically, then it is called as chemical reaction. Physical change is the temporary, chemical change is the permanent. That you will get detailedly in the physical chemistry in the 11th, 12th classes. Okay. That also he has given some few lines here. Now you see here, what is chemical reaction? One reaction example he has given, that is barium hydroxide plus sulfuric acid. It will chemical reaction, it will undergo and becomes the barium sulfate and water. So it is chemical reaction. Means here the substrates are the barium hydroxide and sulfuric acid, but product is the barium sulfate, and next will be the water. So new products came. That is called as a chemical reaction. Physical reaction I told already. Water become the ice become the water. Water will be evaporated. All these things are the physical changes because the no change in the chemical in that one. Now whenever we are seeing the rate of reaction, chemical reaction, whenever we are seeing, it will be one formula is that delta P by delta T. What is P? Product. By the time, by the unit time, how much product is, this is the product. It is the delta product, it is coming within the time. This is the substrates, it becomes the product. How much product is produced in the unit time, it is called as the rate of reaction or velocity of the reaction. Velocity or the rate of the reaction is having one formula, that is the delta P by delta T. Now already we saw inorganic chemical reaction. What about the organic? Organic chemical reactions also. Organic chemical reactions, that is the hydrolysis of the starch as the glucose. What is this? Starch, amylase enzyme used to convert the starch into the glucose in the digestive system. 8th class onwards you are studying. So the enzyme, what it will, that is what it will do. Starch is made up of the glucose molecules. It will be attached one to another. That already we discussed. That bonds will be broken and it will become glucose. That is means starch is by glycosidic bonds it is forming the glucose. Glycosidic bonds it is formed and starch it is coming. And by breaking those bonds it will become glucose. Both are the chemical reactions only. That is only organic chemical reactions. Hydrolysis of starch. Hydrolysis means here, what is hydrolysis? Whenever we are breaking the bonds of the starch, it will take the water. And so for that only after eating, we used to take water because whatever the food material is there, it will become simplified by breaking the bonds. That is the thing, that is a different matter. So hydrolysis of the starch is also a chemical reaction. Now we will see one more thing he has given. How much speed it is. It is coming, active site substrate is sitting and the entire enzyme is making it product, okay, and speed is increasing. How much it will be increased? For that one he has given one example. That is carbon dioxide plus water. Carbon dioxide will be, it will be dissolved in the water. In the natural, it is a reaction will be there. But at that time, in the natural condition, without enzyme in between the dissolution, and it will be only, it will produce only 200 molecules per hour. For 1 hour, 200 molecules of carbonic acid will be formed by dissolving of the carbon dioxide in the water in the natural conditions. Means per hour 200 molecules will be synthesized in the natural automatically. But if the enzyme involved in this reaction, carbonic and hydrogenase enzyme, you will surprise that the speed will increase 6 lakhs per second. For 1 hour 200 molecules with the carbonic anhydrogenase enzyme, if you involve in this one, it will increase 6 lakhs per second, not hour. So per hour it is 10 million times higher. Means you can imagine how much speed it will be. That is the power of the enzyme. And carbonic anhydrogen, this is important for the neat bit. And the figures are also important. And now the thing is, the rate we have told, delta P by T. The delta P by T, when this enzyme is reacting like this, and temperature effect also we saw in the first paragraph. Temperature will be increased, pressure and temperature increases, inorganic catalyst will be increase its efficiency. But up to 40 only, the enzyme will work, then it will become natural, denaturated. Now, whenever we are seeing, there is a thumb rule, general rule, how much the reaction depends on the temperature. Means thumb rule means main one rule, that is either maybe inorganic or organic matter. This is maybe the enzyme reaction or inorganic catalyst, whatever it may be. They will follow this rule. Every 10 degrees centigrade, if you are increasing, every 10 degrees centigrade, it will, the reaction speed will become double. This is most important bit for the need. Every 10 degrees centigrade, it will become double. Actual catalyst means it may increase, just he told, but it may decrease also. Means accelerator and a brake. Accelerator, if you press, the vehicle will go speed. Brake you touch, it will become speed. The speed will be reduced. Both the things will be done. That means increasing the speed of the reaction or decreasing the speed of the reaction will be done by the catalyst. That is the thing. Here, it may be doubles the or decreases for every 10 degrees centigrade. Means here he told, that is it may increase or decrease. Both the things will be done by the catalyst. Biological catalyst, the enzyme. How the biological catalyst work? He has given one example: carbon dioxide plus water in the presence of the carbonic anhydride, it will produce the carbonic acid H2CO3. How much speed it will do? Generally, without the enzyme, it will produce only 200 molecules per hour. But when the enzyme involved in this one, the speed will be increased. How many? 6 lakhs per second. Per second. Otherwise, without it will be 200 molecules. But 6 lakhs it become within 1 second. That is 10 million times it has increased. So that is the power of the enzyme. Now we'll see one more reaction he has given. That is multi-step. Generally, within one step, product won't come in the biological. Means it is continuous steps. That is called as pathway. Majority of the metabolic reactions are the pathways. So in that one, multi-stepped means, for example, glucose plus oxygen, it will become this is the pyruvic acid plus water. In one equation we shown, but to become this one to this one, 10 enzymes are required in the 10 steps. Glucose, this entire thing will come in the respiration glycolysis that you will read in the respiration topic. So, but appearing as one equation, but it is taking 10 reactions, after only it will be formed, that is the pyruvic acid. So, when a multi-stepped chemical reaction, in each step means because 10 enzymes means 10 reactions. Each reaction may need only one type of different types of the enzymes. And finally, end product is the pyruvic acid. It is coming. Now the thing is, if it is a complex enzyme means it is made up of the 3-4 enzymes become one mixture, and that complex enzyme will do the entire reaction. Means all the mixture one it will do. That is step by the catalyzed by the same enzyme complex or different enzymes. But in the glycolysis, different enzymes used to participate. We are going to study it in the respiration. But the thing is, it is this entire thing, 10 different steps, end product is coming. This is called a pathway. What is pathway? when the chemical reaction is taking place, end product is coming by the particular way of the reactions. So from the starting substrate to the end product, so many reactions are there. This entire running path is called as pathway. That is a glycolysis pathway. We used to read there again. Now whenever and 10 enzymes in the pathway. Now you see here. Glucose become pyruvic acid through 10 different enzyme reactions in the pathway. So here, whenever we are seeing this reaction, that glucose becoming the two molecules of the pyruvic acid with the 10 enzymes and 10 reactions. And next, this pathway, now one more thing he has given. That is, the pathway will be extended with one, means now the what you are feeling, glucose formation to the pyruvic acid is the end product. But the end product may be extended for some more reactions. What is this? That also will come in the respiration. But the end product, that whatever he is telling, maybe in particular circumstances it work as end product, or in some circumstances it will be extended to give some other product that he want to tell. Now you see, this pathway will be extended with one or two additional reactions to form metabolic end products. That is, he has given. It will come detailedly in the fermentation, in the respiration, that is after glycolysis. Glycolysis will produce the glucose and to the pyruvic two molecules. This pyruvic acid will enter in different ways, that also you will read very clearly, but that is, if in one circumstances the pyruvic acid will work as the end product of the glycolysis, in some other circumstances, situations, the whatever the end product, that pyruvic acid, it will take one or two steps extra and forms the lactic acid or ethanol. That also detail you will read there. But he has given in the anaerobic condition. Aerobic condition means in presence of the oxygen. Anaerobic means without oxygen condition. If this is formed, that is the pyruvic acid. The pyruvic acid will be converted into the lactic acid in the muscular cells. Means whenever a person is doing the exercise, he will get the pain sometimes because enough oxygen is not supplied to the muscle. At that time, the pyruvic acid, whatever is formed, it will be converted to the lactic acid because of the adequate, not enough oxygen is not there. Means what is the situation? Oxygen is not enough, then it will convert into lactic acid. Now here you see. And next one more example, fermentation. When this is coming lactic, what is the pyruvic acid? Pyruvic acid will be converted into the ethanol by taking two more steps. Those steps also you will read. So depend upon the situation. Now you see. So here, so based on the conditions of different end products, formation is possible. So end product is the, if it is entering into the aerobic respiration, end product of the glycolysis is pyruvic acid. If it is entering into the fermentation, it may be the lactic acid or it may be ethanol. So depend upon the end product, it will be extended. From this one statements it will come. And some of the important, these things he can ask, and he can ask this thing also. And the next will be the what is this one, and this 10 degrees centigrade what it will do, and what it is the, all the things are important. So this is the actual explanation whatever he has given, and it is suitable to create NEET questions also. It is important, you must understand. Based on this one only, next steps you can understand in the enzymes. If you like this video, please give the like mark. And if you want to see all my videos and further the videos, that the enzymes of 2 more videos I am going to make. And from enzymes he is asking minimum 3 to 6 bits they are asking every year. In the last year cancelled exam also 4 here. So that if you want to understand properly, you must know the introduction, whatever these things are very important to understand other things. And their questions can arise from this one also. So if you like, please subscribe the channel and give the like mark to see all my videos. And like marks and these things, what it will do, it will, my videos can reach to other peoples also, because the YouTube used to measure how many likes it came in. See, it is a good one, though it will send to other students. So giving like mark is you are helping the other students also. Remember it. Thank you.